Hello, this is Dr. Paul Cottrell, and I'm recording this on 5/9/2026 at approximately 11:08 a.m. As I mentioned in a previous episode, I wanted to find out if the Andes version of the Hantavirus was clathrin dependent or independent. Now, there are two different types of hantaviruses, the Old World and the New World version. The Andes version is the New World. And with New World hantaviruses, they have clathrin dependency and clathrin independency. While the new, while the Old World hantaviruses are clathrin dependent. So there is an added capability for human-to-human transmission of hantavirus through the fusion and the incorporation of the virus into the cell. And just for a quick overview, the clathrin dependent version, what happens is that you have a receptor on the cell surface that has an adapter protein in the tail, and that's embedded in the cytoplasm. And clath the clathrin will attach to the adapter and create this coating. Now, this coating will stabilize and help to bring the actual virus into the cell, and then dynamin comes in and pinches it. And what happens is then the clathrin coated vesicle will start to become what they call naked, where the adapter and the clathrin comes apart, and then it will end up going into an endosome, and that early endosome will break up the actual shell of the virus and allow for the genome to go into the cytoplasm for replication and transcription. So it's very important to understand that the Andes virus of the hantavirus subcategorization has independency and dependency for clathrin. So New World hantaviruses have an additional capability to actually get into the cell. And this is part of the reason why, but not the only, but a small part of the reason why it is worrisome, because it has different pathways to get into the actual cell, dependent and independent, while the Old World is dependent only. Now, what I want to do is also share my screen for the spike and the receptor. So what we're going to do is talk about the receptor really quickly first. Now, this receptor is a Protocadherin-1 receptor. And protocadherins are really important. Different types of cadherins allow for cells to sense its environment, to bind to other cells, and to create this cohesion, a connection. But other molecules and spikes on a virus can attach to a protocadherin and lead to incorporation into the cell. So this research study that was published in July of 2023 was taking a look at mutating some of the protocadherins in some cells to see if a hantavirus would recognize it. And what they found was is that if you do certain types of mutations, the hantavirus does not recognize the actual receptor. So that's important. But this paper has some good visualization of the spike of the actual protocadherin. So what we want to go over here is that, and there's another diagram that's important to go over first. Where is it? Right here. All right. Like I said, these protocadherins can be used to help to attach cells to each other. So you could have one protocadherin attached to another, and you have two cells attaching. So this is the cell membrane. This is inside the cell right here. This is the extracellular matrix. They're binding the C1 and the C4. And you have another cell membrane in the cytoplasm. So this is how cells can bind together. But a virus can attach to these protocadherins. And specifically for the hantavirus New World versions, they have a strong affinity towards the Protocadherin-1. Now, with the Old World versions, there needs to be additional research because the Old World hantaviruses, they are transmitted from some sort of rodent host or bat to a human, but no human to human transmission. But with the New World versions, like Andes, you have human to human transmission. So you have New World that has this attachment, but you also have a human to human transmission with Andes. Now we're more concerned about Andes because it's human to human transmission. So the Old World doesn't seem to have that strong of an affinity to the Protocadherin-1, but Andes does. And the binding site for the GNGC, which is the glycoprotein N and C, the N is more the head of the spike and the C is more of the stalk of the spike. The binding site is right here on this Protocadherin-1. Now, the binding site, if they wanted to have cell-to-cell interaction, would be approximately here. So if you want cell-to-cell interaction between C1 and C4 cadherins, it would be approximately here. But the binding of the actual hantavirus Andes strain, it is here. And it's at this F8, this F83 and D83 position. That's important. Okay. So if you had cell-to-cell interaction, the molecule for EC1 looks like this three-dimensionally. And then it binding to EC4, it would be right here. All right. So you can kind of see here that you have EC1, EC2, EC3, EC4, 5, 6, 7. And this EC is outside the cell, above the cell wall. And this protocadherin 1 is EC1 through 7 plus part of the molecule that's embedded in the membrane, and then a little bit of the molecule that's actually anchored into the cytoplasm below the membrane. So this whole molecule here would be the Protocadherin-1. Now, EC1 is a location that's above the actual cell membrane on the Protocadherin-1. And the EC4 is that location for the other cell. So you have EC1, EC4, you have EC3, EC2. So this will bind. So they're binding at the EC1, EC4 location, and it happens to be E137, K455. That interaction site will bind two cells together. But if it was going to be a bind for the virus, it can bind here. Now you can see that if the virus is small enough and can get in between cells, there is space to attach when it is attached to the EC1 and EC4 position. It's not being blocked. Because it's not being blocked, there is an opening for that virus to attach to. Okay, and this is a little bit more of a zoom view of this area that we're talking about, where you have F83 and D85, this area right here on the Protocadherin-1 EC1 location. That's where the virus is attaching to on the spike, the actual G and GC part of it. Okay, that's the big takeaway from this paper. Really, the paper is focused more on the mutations, so it would disrupt the hantavirus recognition. But the key here is that we're looking at this location on the Protocadherin-1 for the hantavirus Andes version. And the key location is F83 and D85. Now I want to talk about actually the spike. Now this is a paper that was published in Cell. This was published October 15th, 2020. This is about the glycoprotein. And so it's specific to the GN and GC. Now, just a refresher, I stated before that the spike on the hantavirus is a tetramer. And the tetramer is basically 4 units. And in each one of those units makes up the GN and the GC that we were talking about earlier. And the actual location of GN is here, and the GC is here. Now, we're going to see attachment about right here for the actual cadherin. And you can attach one here and here and here. So you can see that there's lots of ways that the virus could attach to a Cadherin-1. Now, what would it be if we were looking on side view instead of looking down on the spike like the previous diagram? This is the Andes spike, all right, in loop form and also in solid molecular form. And this here, should stop doing that. Maybe I'd stop if I don't know. Okay, so this right here is GN, this is GC, and there are different domains, domain 1, 2, and 3. So this is domain 1, this is domain 3, this is domain 2. And this is the tip, what's called the tip. All right, so we're looking at the tip, and then we're talking about some of this in the actual stem. We'll call this the stem. All right, and this is poking out of this virus here, of this hantavirus. This is the top view. This is the side view. Okay, so we have the head or the tip, and you have the stem. Now, the attachment of this protocadherin, it's coming from this direction, and it would be attaching approximately right here or so. cell. That's the shape, and then there's of the GN and the GC for the Andes version of the virus. And four of these make up the tetramer on the surface of this particle, this hantavirus. Now, what I want to do is I also want to bring up some of the important domains that we need to worry about in terms of, what are the differences? Now, there is something called a beta-3 integrin. That seems to be a receptor candidate for the old world. And then sometimes the old world hantaviruses are also using the beta-1 integrin too, not just the beta-3. Now, for the new world, it's the dominant one is this PCDH1, which we just went over, this Protocadherin-1, which is not used in the old world. And that the old world is generally clathrin dependent. Now, the Andes was first discovered in 1995 in Argentina and in Chile. So the initial identification was in Argentina, and Chile case is reported the same year. Now, in 1993, there was a recognition of a hantavirus pulmonary syndrome in southwestern United States. And the Andes was one of the early New World hantavirus identified in South America, causing the hantavirus pulmonary syndrome, or HPS. The first human isolate documented from a Chilean patient approximately 2002, but the virus itself was identified in 1995. Now, for the old world hantavirus, it was isolated and identified in 1976 in field mice near Hantan River in South Korea. And it was known to cause Korean hemorrhagic fever. And there's a few different versions of it. The Sol virus, the Pum Mala virus, and the Do Brava Belgrade virus. Those are all old world. So the key here for what we're dealing with in terms of our outbreak on the ship, we're dealing with the Andes, which was discovered in 1995. Now, what's key to know about the attachment for Andes is dominate the domain. The primary attachment is this EC1 domain on the Protocadherin-1. And what's going to happen is that It's the key locations on that Protocadherin-1 are the F83 and the D85, which I mentioned before. Those are important to know. Now, I think that would give you a basic overview of the bare bones of what you need to know in terms of what the spike is. That spike is the G and GC tetramer. And what makes up the tetramer is the GN and the GC, and it's shaped like this, right? And the attachment is of the protocadherin from the cell is the EC1 location. Okay. Now I'm going to be doing another video going a little bit deeper in terms of animal models and some other aspects of mutations and gain of function and the genome. So please help support the work that I do. I do all these videos for free. If you could help support the work by sharing the links, making sure that you subscribe to all the channels, all the links are in the description of this video. You can watch me on YouTube, Brighteon, Bitchute, and Rumble, X, GETTR, Substack, Patreon, TikTok. And you can also help support the work by being a paid member to my Patreon channel or Substack channel. You can donate on my website, the-studio-reykjavik.com. You can donate through Stripe, PayPal, or buy me a coffee. Again, all these videos are for free, and please take a look on my website of the great products that I offer. Again, the website is the-studio-reykjavik.com. So hopefully this series on the Hantavirus will give you a better idea of what is going on with the human-to-human transmission, what is unique about the Andes virus and the New World hantaviruses in terms of the clathrin and the attachment to the Protocadherin-1 receptor. Thank you for listening and have a nice day.